Welcome, everyone. This is Dave Iverson. I'm the Michael J. Fox Foundation's contributing editor and the moderator for today's Third Thursday webinar called How Stopping a Sticky Protein May Cure Parkinson's Disease. Just a reminder that you can log in every third Thursday of the month at 12 Eastern, 11 Central, 10 Mountain, 9 Pacific, to hear a discussion about topics of importance to our broader Parkinson's community. And so we hope you'll continue to join us throughout the year and next year as well as part of this series. We also want to acknowledge AstraZeneca for their sponsorship of the 2014 Third Thursday webinars. Throughout the discussion today, and this is a complex, fascinating topic, and we know you will have questions, and so we want you to be able to submit them, and you can do that just by going to the Q&A box at the bottom of the screen and typing in your questions there, and we'll do our best to get to as many of those questions as possible in the second half of today's presentation. And before we get to today's topic, I want to also mention that if you want to download the slides from today's webinar, you'll see a box called Resource List at the bottom of the screen. If you click on that, there'll be a link there. You can open that document then in a new browser window, and you'll be able to save or print the slides there. Okay, so let's get started. Here's what we're going to try to cover today. We're going to go through what alpha-synuclein is and the Lewy bodies that are formed largely out of alpha-synuclein and why that becomes problematic in Parkinson's. We're going to talk then about that relationship that this protein has to Parkinson's disease. And then we're going to focus on how researchers are targeting that difficult protein, alpha-synuclein, with new therapies. And then we'll talk some about how we need to figure out how to measure it better in order to really get at this problem and solve it the way we all hope we can. So let's meet who's going to be participating in our discussion today. We have two leading Parkinson's researchers who are going to be part of our discussion. Kuldip Dave is one of the senior scientists at the Michael J. Fox Foundation. Kuldip, thanks for being part of our conversation today.
Thank you for having me.
And also joining us is Dr. Jeff Cordover. He is the director of the Research Center for Brain Repair and a professor as well at Rush University Medical Center in Chicago. Jeff, thanks for being part of our conversation too.
Always a pleasure, Dave.
So let's get started by first defining what we're talking about. And Jeff, let's look at the first couple of bullet points that we now see on the screen. Title, Lewy bodies are the hallmark of Parkinson's disease, that people with Parkinson's have these clumps of protein, largely formed from the protein alpha-synuclein, that get stuck together and form these Lewy bodies. This is a relatively new understanding, right, about this condition. We've known for a long time that dopamine cells go missing. Hasn't been completely clear why these Lewy bodies really seem to be part of the key, correct?
That's correct, Dave. Well, first of all, Lewy bodies are the pathological definition of Parkinson's disease. So if you don't have Lewy bodies in the brain upon autopsy, you can't be defined as a patient with Parkinson's disease. That's how integral they are to the aspects of Parkinson's. They're really formed from a protein, as you mentioned, called alpha-synuclein. Alpha-synuclein is normally found in everyone's brain in a form that is not toxic, and we're not sure exactly what the normal function of alpha-synuclein is, but we think that it helps facilitate communication between one cell and another. But what makes it different in Parkinson's disease is that the shape of alpha-synuclein changes, and it forms this clump, as you see there in the picture. And it's sort of like a garbage disposal having clumps of food stuck in it, and then you can't clear it. You can't grind up the food, and it causes the garbage disposal to break. And what happens in Parkinson's disease, we think that this clump causes the cell to become dysfunctional and ultimately causes the cell to die. Now, why do we think that this is important and really integral among all the other pathologies in Parkinson's disease? And that really comes from studies of genetics. And there was a landmark paper by my colleague Andy Singleton and his co-workers at the National Institute of Health a number of years ago that showed that the only genetic defect you have is that you make too much alpha-synuclein, you get Parkinson's disease. And that probably is one of the most important pieces of data we have that speaks to how integral alpha-synuclein pathology is in the symptoms of Parkinson's disease.
So before we climb into further why alpha-synuclein goes wrong, you mentioned the genetics, and there may be other reasons why it can go wrong as well that we don't fully understand yet. I want to make sure that I and we all, I guess, are really keying in on the key factor here. It sounds like, they literally gum up the works. This protein that is in everyone, but somehow, for some reason, in some cases genetics, in Parkinson's, this protein that everyone has literally gums things up. Is that essentially what happens?
That is exactly what happens. And by gumming up the cell, lots of different things can happen. So you can have two cells right next to each other in the brain of a Parkinson's patient. And one cell has this Lewy body, this gumming up of the cell, and one cell doesn't. And the cell that does, one of the first things it does is it stops making dopamine. And the absence of dopamine actually what causes the cardinal symptoms of Parkinson's disease. That same patient with the cell right next to it that doesn't have this clump has normal levels of dopamine. In addition to stopping the dopamine production, lots of other changes occur in these cells. So if you have the clump, the ability to clear, to have the garbage disposal clear unwanted proteins is greatly diminished, while the adjacent cell that doesn't have the clump goes along with this process quite normally. So there are many features of normal cellular function that when you get this, that under normal circumstances with normal alpha-synuclein work quite well. When the clump comes, it really stops many of the major cellular functions of the cell, and ultimately it leads to cell death.
And Dr. Dave, in just a moment, is going to sort of talk us through some of the ways we may be able to deal with that clump that scientists all across the world are now working on. But before we get to that, let's go ahead and look at our next slide and just dive a little bit deeper into what we think may be going wrong, because as you suggest, genetics may be part of that. So if we could go ahead and look at the next slide, we'll have a chance, I think, to understand what's going on just a little bit more. So as we've already mentioned, everyone has this protein, and as you were saying, Jeff, there's something that goes amiss, that goes awry, that there's a kind of bad form of this that happens. And tell us what we know and what we don't know, I guess, about why that happens. You mentioned genetics are one reason, but that doesn't explain it for everyone necessarily, right? Is that part of it we still don't really know, why this thing starts to clump up?
We don't know why it clumps up, and we don't know where it clumps up first. So in terms of why it clumps up, we don't think that it's made, in most circumstances, the machinery that makes the alpha-synuclein doesn't make it at too high a level, but we think that it starts to not be cleared normally by certain features of the cell, and so it starts to accumulate. And this accumulation likely causes it to change its shape, we call that misfolding, and become this abnormal structure called a Lewy body. Now,
so it's not just, so Jeff, it's not, it sounds like it's not that people in Parkinson's, it's not like they're necessarily producing too much, but we know that people with Parkinson's wind up having too much. So it's getting backed up, essentially. The production process, it's like a water faucet that's turned on, the water's flowing just like with everyone else, but in Parkinson's, the drain isn't working?
That's correct. And when that drain starts to not work properly and you get this accumulation, that sets off a cascade of changes within the cell that stops it from being cleared normally, and then because it's not cleared normally, it's normal. We have more that's being normally made that's being added to it and added to it and added to it, and so that ultimately leads to the cell degenerating.
Okay, so let's now dig into looking into what's going on in the cell, which will lead us then to some of the possibilities that you and all of your colleagues are looking into to solve this problem. So if we go ahead and look at the next slide, Kuldip Dave at the Fox Foundation is going to start giving us a sense of what's going on. So Kuldip, Jeff mentioned that there's, proteins are made by genes, and in some people with Parkinson's, there's a genetic problem, though that won't explain it in everyone. But begin to tell us how this works. We see on the screen now DNA, mRNA to protein. What are you illustrating there in that particular diagram?
Yes. So what's shown up there is a blueprint for how all of our proteins in our body are made. There is, as you know, DNA in all of our cells, and this DNA produces a message, and which we call mRNA, which is the second component that you see there. And this message is then made into a protein, and this could be any protein in our body. In this case, we're talking about alpha-synuclein, but this is how we get alpha-synuclein protein. So when Jeff earlier mentioned genetics, mutations in the DNA or changes at the DNA level would then affect protein levels. And so that is one way that we could get higher levels of alpha-synuclein in the brain. So
this would then lead, so the next thing then would happen when that process, at least in the genetic cases, goes awry, then you start getting too much, it starts clumping together, and that's when the Lewy bodies start showing up?
Exactly. So as you see on the screen, and as Jeff mentioned, this protein is in everyone. We all have this protein. In disease, however, and there's that big red question mark, we don't know what it is that triggers as Jeff said, misfolding of this protein and the clump formation that happens that eventually leads to Lewy bodies. So that normal function going to it being an abnormal misfolded protein leading to Lewy bodies is a big question right now.
And so what happens then next? And begin to tell us then some of the strategies that are being developed to begin to solve this problem.
Okay. So again, as Jeff mentioned earlier, these Lewy bodies then start or initiate a cascade of bad events that start happening, the end result of which is the death of dopamine neurons. We also think that these Lewy bodies could be released outside of the cell. So that's what you are seeing in the diagram. So synuclein and Lewy bodies are normally found inside the cell, but the idea is that some of that protein also gets to the outside of the cell. And so that toxic protein is available or is present both inside and outside the cell. And so this is what we know, and our strategies to go after this protein therapeutically sort of go after these different aspects. So as the slide shows, the first thing we can do is to mop up the alpha-synuclein that's already outside of the cells. And so this is what we call catching the Lewy bodies on the move.
And Calder, before you go a bit further, I just want to understand something about Jeff. Jeff Cordover, you were saying earlier that alpha-synuclein may be involved in messaging between cells. So is this key to what happens, that this forms and it gets sort of sent out, and therefore it can kind of start going from one cell to the next, and that's why this one strategy is then to sort of catch it once it's out there moving around?
Well, under normal circumstances, in normal people, we don't believe that there's evidence for transferring from one cell to another. But there's this novel hypothesis that has been around the last couple of years, in which we've demonstrated that once you get this clump, that the clump can get outside the cell and then picked up by a neighboring cell. And so one of the things we are, from what we call pathogenesis, for why the brain undergoes degeneration point of view, we think it may not be that we're getting more of these Lewy bodies that causes disease progression, but rather the Lewy bodies are transferring throughout the brain, and actually even from the body to the brain, and then throughout the brain. And this is what may be responsible in part for the progression of symptoms in Parkinson's disease.
And so, Calder, then back to your idea of mopping them up. One strategy then would be it's not so much shutting down or busting up, we'll get to some of those ideas in a moment, these clumps, as it is to keep them contained so that you're sort of keeping them from doing too much damage? Is that what the mopping up strategy essentially does?
Yeah, so it's the theory, as Jeff mentioned, that clumps are released from cells and they're infiltrating other cells, normal cells or healthy cells, to, quote, spread the disease. It could also be another hypothesis is that the disease cells are somehow giving off a signal to the other cells, and then alpha-synuclein in the healthy cells now start to clump and misfold. And this is how the disease is spreading. So if we had a drug that could target this spreading between cells, then we could stop the progression. So that is one way. I think, and I'll go on to the other strategy that the foundation has gone after, is lowering alpha-synuclein levels overall. And so here, as I earlier said, we go from our standard DNA to a message to protein formation. If we can lower protein levels by somehow manipulating or tinkering with that process from going from DNA to the protein, then we can lower the synuclein protein levels overall. And if we do it this way, then if there's less protein to be made, then there would be less chance or propensity to form Lewy bodies in the disease. And so as Jeff was saying earlier, that our cells cannot keep up, our clearance or garbage disposal cannot keep up with the clumps, that if we can lower the production of protein in the first place, that maybe our cells can then catch up in how it clears away clumps of protein.
So Calder, this would be, to stay with the plumbing analogy for a moment, this would be like turning off the faucet, right? Or turning it, reducing the stream, so to speak, so that there's less sludge developing.
Exactly. So if we could just interfere with gene signaling in such a way that we can just lower what's going into that drain, then we would have less gunking up the drain.
Okay, so we've got the mop. The mop is standing by, ready to mop it up if it advances too far. We've got the possibility of shutting off the faucet so that not so much flow takes place. But there are a couple of other strategies as well. So lead us to those.
Yeah, the third strategy is going after the clump itself. And some of you may have heard about scientifically people calling this strategy disaggregators or disaggregation. So as Jeff mentioned, we have Lewy bodies. These are accumulation of alpha-synuclein. And how about just having drugs that bind to these Lewy bodies, these clumps, and just break it up, just liquefy the clump itself. And if we could do that, we could break up the Lewy bodies. Now the cells could function properly because even though there is some signal in the disease process that's making more and more Lewy bodies, if we can just keep breaking them up, then the cells don't really have to. The cells can really function and not die.
So that's sort of the Drano approach, right? You're dissolving the clumps.
Exactly. We're just dissolving it up so that you just get it to pass through, and your cell and your drainage system starts working. The fourth approach is, all staying on that drain analogy, is to increase the garbage disposal capacity of our cells. So Jeff really did a good job explaining how we see our garbage disposal. All of our cells have a garbage disposal where proteins are generated, and then we get rid of them through this garbage disposal system. And this is how we also get rid of abnormal proteins, such as what are found in clumps or Lewy bodies. So if we could increase the capacity of our garbage disposal system, we could then get all of this gunk cleared up quickly and more efficiently. And these are really the four ways to go after alpha-synuclein. I think what's the strength here is that we don't know which one of this approach will work. And this is a multi-pronged approach, and having a more, as we like to say, diversified portfolio to go after alpha-synuclein.
So let's look at the next slide then and see where these four different approaches are currently playing out in the lab and in some cases in clinical trials. And we see the four of them listed here. There's the mopping up strategy, and which we'll describe in a little more detail. There's the breaking up of the clumps, and that's what I was calling the Drano approach. There's the making the garbage disposal work better, the clearance idea. And then the last one is what you were talking about, which is sort of turning down the faucet, reducing the amount. Before we talk about the where each of these stand, and you can see on the graph there, some of them are further along in the laboratory and clinical trial process than others. Jeff, let me bring you back in and just underline, if you would, the importance of what Kuldip was saying about having a lot of possibilities, that just like with your own clogged brain, you may need alternative approaches. That's why this is important, right? We don't know which way it's going to be that will actually clear the drain.
Exactly. And, let me give you an example of where there are many drugs that are quite effective for a number of different things, but they only work in a very small therapeutic window. And so we have to understand that all of these may work in the laboratory to some degree, but which of them will be effective and safe? So I want to just point attention to the protein synthesis one, the last bar there, because although it's an excellent idea to reduce the amount of alpha-synuclein, I remember when I bought my first house, and I turned on the garbage disposal without having any water running through it. And the person said to me, don't ever run your garbage disposal with no water. And so if you drive protein synthesis down to very low levels, you can actually get enhanced pathology. So some of these may work, but they may only work in a sweet spot of dosage. And that's also part of the problems going forward. So, but from my perspective, I really like the idea of increasing clearance, because that should be ultimately not only effective but also very safe. And the ability to disaggregate, is also a very exciting therapeutic potential in the future. Recently, I just saw Kuldip and I were presenting at the Michael J. Fox Therapeutics Conference, and the first presentation that I had seen for immunotherapy, which is now in phase 1, just completed phase 1 clinical trials, while we can't make any statements about whether they're efficacious or not, the one thing that was quite promising is that it appeared to be very safe. So having multiple approaches, looking at both efficacy and safety, I think is a very positive path moving forward, because there are multiple ways to address the same problem.
So let's stay with that one at the top, the mopping up one, first, Kuldip, and it also uses the word immunotherapy, and this has been sometimes described as an alpha-synuclein vaccine, which is sort of a hard concept to get your head around, the idea that you could actually have a vaccine against alpha-synuclein. But that's the one that's actually, as Jeff was saying, is the furthest along. It's gone through phase 1, the safety trials. Now it'll be starting to be examined for whether or not this can actually work. But describe the idea, if you would. Is this the same basic idea as when you get a flu shot?
Yes, it's very exciting. And so the way it works is you, just like flu shot, you go in and the nurse or the doctor injects you with a dead, inactive form of flu virus, so that in the event, in a couple of months, if you're exposed to flu, that your body has a better way of mounting an antibody response when you are exposed to flu. So the same idea is in this trial that's ongoing right now for synuclein. The company has figured out a way to inject inactive alpha-synuclein in the body, but this alpha-synuclein that's injected is the misfolded version. As Jeff was saying earlier, it's the abnormal version of alpha-synuclein. And so your body now will generate an antibody response, as it does for anything that we are exposed to. Our immune system has evolved to fight bad viruses and bacteria and proteins that go into our body by making antibodies. And so this is essentially the concept for the synuclein vaccine, that an inactive, bad form of alpha-synuclein is injected. Our immune system will now mount an antibody response, and now those antibodies will presumably get to the brain and mop up the toxic form of protein that is outside of the cells.
So we know now that from the early, this is now in clinical trials, this is one that's actually being tested in people, and we know so far that it's safe. The next thing, obviously, is to establish in phase 2 and phase 3 whether or not it seems to actually work. How long will that be, Kuldip, before we have a real sense of whether or not this particular strategy, the mopping up strategy, vaccine strategy, may actually do what we want it to do?
Yes, so I think we're looking at another, I would say, 3 to 4 years for a phase 2 study to be completed and inform us as to what dose of the vaccine we need to give, how many doses need to be given. This is, as Jeff said, very exciting. The first study showed that it's safe, and these patients were followed for 12 months after 4 injections of the vaccine. But now we need to understand, in a real case scenario, how many times the vaccine would need to be given, how often, how long between the 2 vaccinations, that it would be safe, but also reducing synuclein pathology in the brain. So These are the type of questions that will get answered next, now that we have established safety, what that, as Jeff mentioned, therapeutic window is for this vaccine to work. So we're looking at it's not going to be tomorrow or over the next 2 years. This is going to take at least 4 to 5 years for us to know what it looks like in phase 2 efficacy study.
And Jeff, you mentioned that you were particularly intrigued by the possibilities of the next two, which are not as far along. One is sort of breaking up the clumps, what I was calling the Drano approach, the other being increasing the cell's own garbage disposal capacity. Just sketch out briefly, and then we want to start taking all of our questions, because we have this, by the way, there were more people registered for this webinar than any in the history of these webinars. It shows, I think, how much interest and fascination there is with all of this. So we want to get to your questions in just a minute, and also talk about some of the problems involved in measuring alpha-synuclein, which are part of the challenge. But Jeff, if you would, just briefly, with these next couple of approaches that I was describing that you're particularly enthusiastic about, how far along do we still have to go? What do you see as being both the promise but also the challenge before we'll know whether or not those may be the candidates best suited to do the job?
Well, with regards to the disaggregation and the clearance mechanisms, there's still, we got to show efficacy in animal models, and that we also need to get a better understanding as to what stage of the disease that we try and mimic in animal models would be best to apply these approaches. So if we took a very late stage Parkinson's patient where they had so many of these clumps, would we really be able to, and also we have so much degeneration that's taken place, would really breaking up the clumps do anything significant? But if we try and model for early stage Parkinson's disease, where presumably we just have fewer of these clumps, and we're able to clear them or disaggregate them, that that may have very, very powerful effects therapeutically. The reason I like the disaggregation and the clearance issues more so than the protein synthesis, reducing the synthesis of alpha-synuclein, is because alpha-synuclein is in the brain normally. And there's a reason that it's in the brain. So there is some concern that if you're taking a normal protein and you stop making it or shut down making it, that could make things worse. All these things we can test in animal models and determine its safety and whether we can go into clinical trials. But I think that those are slightly less far along.
And Dave,
can I
add something?
Go ahead, Calder. Yes, go ahead.
It's important to also make the point that therapeutics that are targeting alpha-synuclein are really going after modifying the disease or preventing or stopping the progression of the disease. So despite of these 4 different approaches, general approaches to going after alpha-synuclein, we're not talking about prevention. The idea is that people have Parkinson's disease, and we need to prevent this disease to progress or even stop it. And so all 4 of these approaches, and they have their pluses and minuses, or positives and negatives, as Jeff mentioned, they are going after the disease pathology and the process of the disease. So the idea is to affect people with Parkinson's disease, not something that's happening as a preventative therapy. It could be, as we find out maybe many years down the line, could be used as a preventative therapy. But right now, we're looking at this, and why so much interest in alpha-synuclein is because we think we can modify the disease.
Well, it's a great point and worth underlining, and I guess and additionally we should make clear that what's so exciting about this is its possibilities for actually slowing down or stopping the disease progression. It's not just giving us a better dose of Cinnamon. It's not just giving us a better pill that helps with symptoms, as useful and important as that is. This is going beyond just symptomatic treatment. This is beyond just sort of helping someone contend with their Parkinson's symptoms. This is actually getting at something that would slow down and hopefully stop disease progression, and that's why, of course, there's so much energy right now in the scientific community focused on this. Let's take some of your questions now. There's another thing we want to get to before the end of our webinar where we're going to talk some about an additional challenge, which is the problem of actually how do you measure alpha-synuclein? How do you know how much is there? And how do you know then whether or not the treatment that you're trying out is actually working? But before we get to that, let's take a couple of your questions. Jeff, here's one that I'd like to throw out to you. One person has written in saying, I've been told that it will take another 6 to 10 years, and you both have said that, before the vaccine might actually be out there. Why does all of this take so long? Will it actually be a success? My own neurologist is somewhat skeptical about this. Let's speak a little bit, Jeff, to the challenge of drug development, that people are antsy. This is not a time-neutral disease. It's frustrating when we hear that it may be, even with something as far along as this vaccine idea now entering phase 2, it may be another half a dozen years before it actually could be out there.
Yeah, so there's a process by which new drugs get to the clinic. And the first thing we want to make sure is that it's safe. And that's why that phase 1 trial is what we call a safety and tolerability trial. And it's done in a small number of patients. And while we may look for efficacy in some regards, really the only thing we can tell is that it's safe and effective. That it's safe and well tolerated. So that takes about a year or two to start, perform, and analyze. Then you go to a phase 2 clinical trial, and that is a efficacy trial. And it's done under most circumstances in a blinded fashion, meaning that the patient doesn't know what they get. The doctor who's analyzing them doesn't know what they get, and until the trial ends when the blind is broken. And that's done in a larger number of patients. Probably I'm guessing here, but I would guess that a vaccine trial would probably be between 50 and 100 patients in each group. And so you need the time to recruit them, to do the study, and assess the data. And then unless the data is overwhelmingly strong, you then even have to go into a larger study, a phase 3 study, where you probably have 300 to 400 patients per group. And so all these things just take time. And although I want to assure all the patients that the scientists are as anxious, I'm not going to say we're as anxious as you are, but we are anxious in trying to push the speed of this process forward. It needs to be done in a safe and in a valid way so we can move forward. There are many cases in medical law for which these things were not done in a systematic fashion, not done in a well-controlled fashion. And ultimately, many, many years later, people ended up being treated and even operated on for drugs that really had or treatments that had no efficacy. We don't want that to happen under these conditions.
And it's also why you can't place your bets on just one possibility, right? Because it takes so long, you want to have a lot of different options out there. Otherwise, it's going to be forever. If we just had one and that doesn't work, then where are we?
Exactly. And the other thing that's quite important for people to realize is that it's important for Parkinson's patients to participate in these clinical trials. Often it is very difficult to fill the number of patients that we need to do a proper study. So I encourage people to go to Fox Trial Finder and participate in clinical trials, because without your participation, we'll never be able to get to the finish line.
And if you go to the Q&A tab at the bottom of the screen and click on that, there'll be some resource list and some reference links, some of which are about different trials that are now recruiting people and other opportunities. And as Jeff mentioned, foxtrialfinder.org is another way you can go about getting involved in clinical research. Colton, here's a question for you from Mario, which is, what do we understand about the normal function of alpha-synuclein? We've touched on this a little bit already, but it is one of the interesting problems, and as Jeff suggests, it may be one of the concerns we have about shutting it down altogether because we're not entirely clear on what it actually does, and it probably is there for a reason.
Yes, just right. We do not fully understand the normal function of alpha-synuclein. We have some ideas based on the research over the last 15, 20 years around, as Jeff had mentioned, communication between cells. We have these neurotransmitters, chemical substances that are released between cells, and this is how brain cells talk to each other, communicate with each other. And it's been shown that alpha-synuclein may be involved in this neurotransmitter regulation, or how these chemical substances are released from one cell to another. So this is a lot of our efforts at the Fox Foundation are also trying to understand the normal function of alpha-synuclein, because if we were to make therapies against this target, we need to understand what we are targeting. So I couldn't agree with Jeff more that having that understanding of the normal function will really inform us better of how could we go after this protein.
Question, Jeff, for you from Shane, who writes in, is there any difference in the level of these Lewy bodies between young onset Parkinson's and older onset Parkinson's? Tough question to know because we can't really see what's going on inside the brain, but do we have any sense of whether or not that progression is different?
We really don't, and we just have inferences from small numbers of autopsy studies. There doesn't appear to be a huge difference in either the number or distribution, with the exception of those patients who end up developing dementia. And those patients who develop dementia tend to be, or are really, the older population. And they tend to get Lewy bodies in the most superficial regions of the brain called the cerebral cortex. And so that's really the only clear increase that is age-dependent. So if you look at the substantia nigra in a young onset or an old onset, they look relatively similar. But the progression to other regions, such as the cortex and even the striatum, where dopamine is secreted, that in those areas they are increased in older people, but not necessarily in older onset Parkinson's disease.
But this takes us a little bit into the other area that we want to talk about. And so let's go ahead and take our next slide, because a lot of people are also asking questions about this. Because as you were mentioning, Jeff, right now we can only make those assessments after someone dies and donates their brain to science, and you do the pathological work and you can find these clumps. And that's a big concern, right? Because right now we don't have a way to measure someone in the live patient. Just describe a little bit about why this is so important, the ability to measure alpha-synuclein and how far along, I guess, we are in being able to do that.
Well, this is a very strong initiative for the Fox Foundation and also for all researchers in Parkinson's of Parkinson's disease. It is essential that we find a marker. There are a number of different types of markers. Some could be markers in blood, some could be markers in cerebrospinal fluid. My group and many others are trying to work at imaging the alpha-synuclein using scanning techniques to see alpha-synuclein in the brain. First of all, we'd be able to tailor synuclein-altering therapies specifically based upon the expression of alpha-synuclein in a particular patient if we knew where and how much that patient had. And also we'd be able to evaluate whether the therapeutic strategies that modify synuclein are actually doing that. And that would be particularly important. And then finally, we know that alpha-synuclein occurs well before anyone gets symptoms. And so to be able to say, yes, this person will ultimately get Parkinson's disease, and we can intervene therapeutically, would ultimately be the most powerful therapeutic strategy to go after.
So, Jeff, does that mean that we think these clumps are showing up well before someone actually experiences symptoms?
Yes, there is. So there's kind of a conundrum. So there is a entity called incidental Lewy body disease, and that is when at autopsy you see the same Lewy bodies that you see in Parkinson's disease, except the patient has yet to demonstrate the symptoms of Parkinson's disease. And we think that that's a pre-Parkinsonian state. So yes, we think that these Lewy bodies occur well before the patients get symptoms.
And so if we could catch that then and maybe apply some of these strategies early on, we would be that much further along at really almost stopping the disease before it even starts. Let's go ahead and look at the next slide if we can, because in this slide we're going to get a sense of sort of all of the places that we're developing ways of trying to develop that better marker, that better measurement. And Calder, walk us through this. We're starting out in the brain, as Jeff was describing. One of the ideas would be able to image it, but take us through this and the other places and other ways that you're trying to develop those measurement techniques.
Yes. So as Jeff mentioned, Fox Foundation has a major initiative around trying to develop an imaging compound, an imaging drug that would allow us to put patients in a scanner and look at their alpha-synuclein pathology. But while we are developing that, the idea is to use other parameters in the body that would predict what's happening in the brain. And so the one that's very closely connected to the brain is our CSF, or cerebral spinal fluid. So this is the fluid that bathes our brain and spinal cord, and is where we think that when synuclein is released outside of the cells, that it may end up in this CSF fluid compartment. And so and we know we're able to measure alpha-synuclein in CSF. So the idea is that if we can measure what is happening in the CSF to alpha-synuclein, that may tell us what may be happening in the brain. But then moving beyond brain and CSF, as Jeff mentioned, alpha-synuclein is found in other parts of the body. And maybe those other parts of the body may tell us indirectly what may be happening in the brain. So the other areas where we know synuclein is found is our colon, or our gastrointestinal tract. It's found in the skin, found in our saliva or salivary glands. It's found in cells of the body. It's also thought of to be found in the retina. And of course, our blood has a lot of alpha-synuclein, so our blood cells carry a lot of alpha-synuclein. Now, the issue here, and there are a couple of issues here. One is, how do we measure alpha-synuclein? Do we have specific tools to measure alpha-synuclein? And are those tools really measuring, as Jeff was mentioning earlier, normal alpha-synuclein, or is it measuring abnormal or misfolded alpha-synuclein? So that's something the Fox Foundation is funding right now to standardize how we measure alpha-synuclein outside of the brain. And then two, once we are able to measure it in a standardized way, how do we correlate it back to what may be happening in the brain of a Parkinson's disease patient? So that's really the challenge that faces determining biomarkers, as Jeff was saying, for alpha-synuclein.
And I want to get and use some of our remaining time to take some final questions in just a second. But Jeff Cordover, as you look at this slide, is this in some ways analogous to what we were talking about before, that we need many different approaches to deal with the alpha-synuclein problem? We also want to develop a variety of approaches perhaps to measure it, because we're not entirely sure what's going to work best. It might be something as simple as a blood test or a skin biopsy. Maybe there's something with imaging. But we want to explore, is that sort of the same principle at work here, that we want to have a variety of arrows in our quiver, so to speak?
Yeah, until you know what works, you don't know what works. And so because each of these takes such an extended time to develop, you don't want to go down one silo, found that it's the wrong silo, and have to work your way all the way back and start anew. So I think it's important to try a variety of these biomarkers and test a variety of these biomarkers, and I'm confident that in the not-too-distant future, at least one of them will turn out to be positive. And when we do that, that will be a major advance in Parkinson's research.
So a couple of more questions, and then we'll get to some last comments. Jefferson asks, do DAT scans show clumps of protein? Jeff, I assume the answer is not. That's why you're looking for another imaging system.
Yeah, DAT scan measures what we call the dopamine transporter. So that's a protein that when dopamine is secreted by a cell, when it's finished its business, it grabs it and pulls it back into the cell for repackaging and reuse. And so all it tells you really is levels of dopaminergic, it's a marker of dopaminergic function, and really does not in any way address the issue of alpha-synuclein.
Cecilia wants to know, whether these protein clumps are only in the substantia nigra, which is where dopamine production happens, or elsewhere in the brain. We've touched on this a little bit already. Jeff was describing that they are, in fact, found elsewhere in the brain. But that's part of what this slide is also interesting about, is you're saying these clumps show up not just in the brain, they show up in the colon and GI tract, which may be part of the reason why so many people with Parkinson's have constipation problems, right? So it's sort of these clumps of alpha-synuclein occur in many places and really kind of match up with symptoms of the disease. Would that be correct?
Yeah, more or less. I think the idea is when you have clumps and it forms this pathology in certain area of the brain or certain area of the body, that is going to affect the function that portion of the brain or portion of the body is having. And so if you have aggregation of alpha-synuclein in the colon, that may then disrupt our normal colonic function, and so something like constipation or other GI issues could potentially be driven because there is clumps forming in the colon tissue. So that's really the idea. Yes, and to answer the question from earlier, we do indeed find alpha-synuclein pathology outside of the substantia nigra, in the cerebral cortex, in other areas, hippocampus, which is what Jeff was mentioning. As the disease is progressing, we are seeing more and more of these clumps in other areas of the brain that start to affect both the motor issues that are seen in Parkinson's, but also non-motor issues that are seen in Parkinson's.
In other words, as the disease advances and there are problems later in the course of the disease that might range everything from, problems with cognition or dementia or voice or any number of things, those kind of track with where these clumps are being found elsewhere in the brain and the body?
Yes, that's what we think.
Yeah. Great question that I'd love to get an answer on. This person writes in, so why doesn't our body already produce antibodies if we have too much alpha-synuclein? Jeff, you want to take a flyer at that?
Yeah, because it's, I don't know the answer to that. But I know it doesn't. I mean, it probably views it too much as self. Because it's not being seen as so much as the as a foreign body by the individual because it's still the same alpha-synuclein of the individual. So it's unlikely that it would mount an antibody response by itself.
Yeah,
and here's another. Yeah, go ahead, Calder.
Sorry, and a couple of researchers have looked into that. So this is a very good question, and what we call auto antibodies, and we have not been able to find them yet. That doesn't mean that they're not produced, but that's a very relevant question.
And Jeff, a question about whether or not these therapies that we've been describing would help with other forms of or related forms of movement disorders. Progressive these are referred to as Parkinsonisms or Parkinson's plus, but things like multiple systems atrophy, progressive supranuclear palsy. Would any of these therapies work potentially with some of those other conditions?
That's an excellent question. Now, not progressive supranuclear palsy, because that's the problem. There is a different protein called tau. But for multiple system atrophy, we and a number of other groups are just starting to get very interested in synuclein therapies for that, because the difference between multiple system atrophy and Parkinson's is that in multiple system atrophy, the synucleinopathy, the synuclein clumps, occur not in neurons but in glial cells, a different cell type in the brain. But still, if you're able to deplete alpha-synuclein or disaggregate it, that should help as well. And there is a strong effort going forward for that. Lewy body dementia is a very big problem without a therapeutic solution at the moment, and that's Lewy bodies in the cortex of patients. And so, again, these synuclein-altering therapies should be efficacious for those populations as well.
That's incredibly exciting, I think, because with those conditions that you're mentioning, there aren't even good symptomatic treatments right now.
That's correct.
Right?
One of the regulatory issues that's difficult with Parkinson's is that there are a number of therapies that are available for patients with Parkinson's disease. And so the regulatory agencies are hesitant to allow big therapeutic interventions. While for these diseases, which have no therapies and they are very horrible diseases, the regulatory agencies may let us get in earlier in these patient populations.
Let's go ahead and take a look at our last slide and just kind of sum up in the last couple of minutes that we have. So here's where we are. We know that right now we have a couple of things that are advancing in clinical testing. Those are the so-called mopping up strategies, the ways that Calder and Jeff were describing of kind of containing those alpha-synuclein clumps that are already in the move, the idea of a vaccine. There are more drugs that are poised to enter clinical testing in early 2016. We've heard Jeff describe the work that's going on with imaging and other types of efforts to understand what both the normal function of alpha-synuclein is and why it goes bad, other ways of trying to improve our measurement. So a lot is going on. So just a brief last comment from each of you, please, about sort of how you see the field and how, I guess, central you both think this is to solving the riddle of Parkinson's disease. A quick last comment. Calder first and then Jeff.
I think it's an exciting time to go after alpha-synuclein, coming at it from three different ways. Biology, where we are trying to understand what that bad alpha-synuclein is. Why does synuclein go awry? What is that signal that makes alpha-synuclein to misfold and aggregate? And also understanding the normal function of alpha-synuclein. So this one-pronged approach is understanding biology. Second-pronged approach is therapeutics, where we have a diverse or varied approach because we don't want to put all of our eggs in one basket. And then three, biomarkers, where if we can measure alpha-synuclein in the brain and outside of the brain in different components, that we would have a better way of understanding if our therapeutics work. So using this three-pronged approach, we can really move this field forward, and as we go after a drug that can stop or slow the progression of Parkinson's disease.
And so for Jeff, for researchers, this is kind of the bullseye right now, right? I mean, this is where sort of so much energy and attention is being focused.
Exactly, and as I mentioned to you previously, David, at another venue, my chips are all in here. I know that if a patient genetically has too much alpha-synuclein, they get Parkinson's disease. And if they even have more than that, if they have a 3 times the gene dosage, they get Parkinson's disease with dementia. So there's even a dose dependency here that from a person who studies the brain and why it degenerates, that is extremely important. And I agree with everything that Koldeb said. We have multiple prongs to address it. We're developing biomarkers, and I think the other thing is that these approaches are not meant just to help those cardinal symptoms of Parkinson's disease that are mediated by dopamine that we already have therapies for. It's meant to also dramatically impact all these non-motor features of Parkinson's disease that are incredibly problematic, that involve things like gait and dementia, for which we have no viable therapies at the present time.
I really want to thank both Jeff Cordover and Koldeb Dave for participating in our discussion today. It's been, I think, incredibly enlightening, and I think gives us all a deeper sense of why this research is so central to our solving the problem of Parkinson's disease. We'll be sending a link to the webinar on demand to listen to again or share wherever you'd like. There'll also be a survey link in that email. And we hope you will share your thoughts on today's presentation so that we can continue to do our best to refine these as we go forward. Last, we'd like to ask you to mark your calendars for our last Third Thursday webinar of the year, which will be on December the 18th at 12 noon Eastern. In that one, we're going to try to recap all the progress that's being made, we think, and our greater understanding of Parkinson's that happened this year and where we hope to go forward in 2015. So just go to michaeljfox.org/webinars for more information about that. Thank you all for participating. Thanks, everyone, for signing up for the webinar today. I'm Dave Iverson, and we'll see you next month.